disclaimer for the viewers.
Let us see the characterization of the Niosomes. So the Niosomes are characterized for the morphology, size, polydispersity index, number of lamella, Zeta potential, encapsulation efficiency, stability, and the drug release. So size and the morphology of the Niosomes, it can be determined by using the dynamic light scattering, that is the DLS, scanning electron microscopy, transmission electron microscopy, freeze fracture replication electron microscopy, and cryotransmission electron microscopy. So these are the few techniques which are widely used to get the idea about Niosomal size and the morphology. And the Zeta potential, surface Zeta potential of Niosome can be determined by using the Zeta sizer and the DLS instrument. The surface charge of Niosome plays an important role in the behavior of the Niosome. In general, charged Niosomes are more stable against aggregation than the uncharged vesicle, and hence there is a need to measure the Zeta potential of the Niosomes. Then bilayer characterization. The number of lamella can be determined by the AFM, NMR, and small angle X-ray scattering, that is a SAX for multilamellar vesicles. The membrane rigidity of the Niosomal formulation can be measured by means of mobility of fluorescence probe as a function of temperature. The microviscosity of Niosomal membrane is determined by the fluorescence polarization. High fluorescence polarization means the high microviscosity of the membrane. In the case of fluorescence polarization, bilayer thickness can be characterized by using the latter method together with the in situ energy dispersion X-ray diffraction. So this is about the bilayer characterization of the Niosome. Then the encapsulation efficiency of the Niosomes. So the encapsulation efficiency is defined as a portion of applied drug which is entrapped by the Niosome. How much amount of drug it is enclosed into the Niosome. So unencapsulated free drug can be removed from the Niosomal solution using the centrifugation, dialysis, or gel chromatography. So these are the few techniques which are used to remove the unencapsulated drug. After this step, the loaded drug can be released from the Niosome by destruction of the vesicles. The Niosomes can be destroyed with the addition of 0.1% triton X100 or methanol to the Niosomal suspension. The loaded and free drug concentration can be determined by spectrophotometer or high performance liquid chromatographic. So this is about the encapsulation efficiency. Let us see the stability of the Niosomes. The stability of the Niosome can be evaluated by determining means of the vesicle size, size distribution, entrapment efficiency, or several months storage period at a different temperatures. During storage, the Niosomes are sampled at regular intervals of time, and the percentage of drug which is retained into the Niosome is analyzed by spectroscopy, HPLC, and methods. So these are the few methods which are used to characterize the Niosome. Apart from these methods, there are so many several other techniques are there which are widely used to characterize and evaluate the Niosome. So we have discussed the morphology, size, polydispersity index, number of lamella, Zeta potential, encapsulation efficiency, stability. Let us move towards the drug release. One often applied method to study in vitro drug release is based on using the dialysis method. So here for the Niosomes, we are using the dialysis tubing method. So the dialysis bag is washed and soaked with the distilled water. After 30 minutes, the drug is loaded. Niosomal suspension is transferred into this bag. The bag containing vesicle is immersed in a buffer solution with a constant shaking at 25 degrees Celsius or 37 degrees Celsius. A specific time interval, samples were removed from the outer buffer, that is a release medium, and replaced with the same volume of fresh buffer in order to maintain the sink conditions. The samples are analyzed for the drug content by using a appropriate method, that is spectrophotometer or the HPLC. So simply here we are using the dialysis bag. In the dialysis bag, we are supposed to place the Niosomal drug loaded Niosomal solution, place this dialysis bag into the part of the, that is a release medium, which is having a temperature 25 to 37 degrees Celsius, and after predetermined time interval, we have to supposed to remove the sample and analyze it for the drug. So this is about the drug release. So herewith we have finished the characterization of the Niosomes. Let us see the applications of the Niosomes. Niosomes are widely used in the gene delivery, drug targeting, antineoplastic treatment, Leishmaniasis treatment, delivery of peptide drugs, studying immunoresponse. Then it is used for the carriers for the hemoglobin. Then it is used in the transdermal drug delivery system. It is used for the ocular drug delivery system, and it is used for the cosmetics. There are few cosmetic preparations of the Niosomes are available in the market for the aging. So let us see the summary. So we have discussed the introduction to Niosomes, then advantages and disadvantages of Niosomes, structure and components of Niosomes, then methods of preparation of Niosomes, then the drug loading methods, that is active loading and passive loading, then characterization of Niosomes and applications of the Niosomes. These are the few references which I have referred for the presentation, that is advances of non-ionic surfactant vesicles and their applications in drug delivery, Niosome, a future of targeted drug delivery system, the Niosomes as a nanoparticular drug carriers, fundamental and recent applications, then the Niosomal drug delivery systems for ocular disease, recent advances and future prospects. Thank you very much.